The word Scienion, we always get this confusion. Scienion is a very difficult word to pronounce, actually. It's combined two words. It's science and vision, hence Scienion. And we tend to enable life science. I hope I can show you how we do that. Multiplexing by Picoliter dispensing. Sounds a little bit weird. Actually, it is even more weird than it sounds. We've heard a lot about, in the previous talks, about the personalized medicine, traditional laboratory detection methods, and the trend is actually going towards smaller devices, smaller test instruments, point-of-care diagnostics is a very important hit word here, towards personalized medicine. And I'll try to talk a little bit about this in more detail. General outline, talk a little bit about the development of point-of-care diagnostics, field of applications where we can use that, the concepts for a delivery platform, because if you have a small device used for a point-of-care diagnostic tool, you need a device to actually put these drops, put your chemistry on these small devices. So therefore the small title, How to Get Tiny Drops. Later on, some highlights on this delivery technology and customer examples, which hopefully will be very interesting. So let's start with the first one. Going a little bit back in time, diagnostic actually starts a long time ago. As you see here, it was a little bit more crude that day. Of course, nowadays it has become much more sophisticated. Important technologies has been added. Microfluidics has been a major step forward. Rapid test diagnostics, because sometimes we don't have enough time to wait for several days for the results, so we need a very fast result in best time. Therefore, rapid is a very important hit word here as well. Integration of arrays in microflow cells, so you can automate the whole thing. And multi-analyte technology in order to have your test multiplexed, so not only one answer, but several answers for several markers, which eventually leads to the high-definition diagnostic. The trend actually goes a little bit from the usual traditional laboratory tests towards the point-of-care diagnostic, which eventually and hopefully the patient should do in his home or at least at the patient's bed, which eventually leads to personalized medicine, because when we can test and monitor the ongoing treatment, we can actually adapt the medication in a better way. It's a growing market, so within the next 8 years, well, it started 2 years ago, this is a little bit old statistic, it should be raised by about 5 billion, which means by the time I'm done with my talk, we've already spent about $100,000 in this field. Fields of applications are very various. The only problem here is actually who pays for it. And therefore, most of the examples that you see are more in the high life kind of application, where you have allergies, food intolerance, food screening. A very important and very well-known example is food screening, especially for milk, where they screen the milk for antibiotics, because if you have antibiotics in the milk, you can't proceed with the cheese and stuff. Therefore, this is a point of care, because it has to be done at the time of delivery of the milk to the factory, and there is no time to delay. Drugs of abuse, of course, also a very important application. Coagulation, where the patient can actually monitor his warfarin or whatever coagulant dose he's taking. And sepsis, also in sepsis we don't have enough of time. Sometimes 3 days waiting for the result of a blood culture might be good in results, but the problem is the patient is dead by then. So it's better to have a very quick, very fast recognition method. MALDI is being used for this. Unfortunately, not everyone has MALDI in his bathroom, so some new technologies evolve here as well. And of course, the monitoring of the outcome of a personalized treatment is very important. So eventually, we in the first stage, test the patient for personal genomics, whether he has mutations or whatever. Cytochrome P and so is a very important factor there. See which medication actually fits to this person, give him the medication, and while he's being given the medication, scanning and monitoring for the proceeding of the treatment and adapt if necessary. So this altogether brings us to the requirements of these point-of-care diagnostics. So they have to be easy to use. In German language, there's a very nice word. It's called Chefarztsicher, so which means that the head of the clinic can actually use it by himself. There should be no need for additional sample preparation, because we want to have it fast and easy to use. Fast results. We don't want to wait too long for the results. And very important thing, multiplexing. So instead of having one result for only one parameter, you want to have multiplex parameters. And the major benefit here is that you have a very small sample amount and you get a lot of answers. Of course, compact. You want to carry it around if possible. Low price, this is very important nowadays, and it should be adaptable for high throughput production. Eventually, if it takes 2 days to produce one of these tests, then of course it will not be economically feasible. And actually, those last 4 items are the reasons why I'm here, representing Scienion. The concepts for a delivery platform. So as I already said, there is a lot of miniaturization taking place. So miniaturization is the actual drive behind the new diagnostic tools. And this is the most important ongoing thing in the point of care testing and personalized medicine. It should be cost saving and eventually compact. And as you can see here, a number of examples are which I'll go in detail in my talk, and you can see why it should be so miniaturized. The core technology used for that, I'll talk about this right now. There's a number of ways how to actually deliver a substance from your source plate towards your target. You can either use these contacts, so you just dip into your source plate and then you dip onto your target where your sample is supposed to go. I'm not talking about this patient sample, I'm talking about the basic antibody or antigens that you want to fit onto your test. So you use these dipsticks or these pins actually. The problem here is you don't have a volume control about this, so you don't know how much of your antigen actually went onto your substrate. A lot of times you have donut effects. As you see here, the deposition most of the times is not very precise. You can't dispense into cavities, which is a very big backdraw, because most of these tests are now being done in MTP plates, so multi-titer plates, so you actually need to go inside the 96 or 384 well. And of course, you're touching the surface. And with biosensor applications, this might be a little bit tricky. Or nitrocellulose. Also, if you touch the nitrocellulose, you might get a ring, and this might impair the detection. So the solution is a non-contact spotting technology. So what you see here is actually a tip, a glass tip of piezo dispense capillary. I'll describe that in a second. Benefits, here you have a clear drop. This drop can be monitored, and therefore you can control the volume, measure the volume, control the volume actively. Since you're not touching the surface, you don't harm the surface, and you have very accurate dilution series because the stability of these drops is extremely high. So we have CV values of below 0.5%. Yeah, 0.5. Amazing. So to describe how this actually works, this is the dispense capillary as it's being used in the machine. It actually consists of a glass capillary which is surrounded by a piece of piezo element. This piezo element, if we contact it with electricity at the 2 poles, it will contract and a sound wave will go through the glass and eventually hit out a solution. This is usually done at a frequency of 500 hertz, so a lot of spots if necessary. And due to the various sizes that we can produce, we can have different volumes dispensed, ranging from 80 picoliters, 50 picoliters sometimes, to 800 picoliters per drop. So this is extremely small, and you'll see why. Since we can control the electricity that we provide to the piezo element, we can actually control the speed and the volume of the drop. If you give it more electricity, so more voltage, eventually it will fly much further, it will fly with a stronger speed. This eventually will have an effect on how much your drop actually spreads on your surface. So you can control the spot size on your surface by controlling the speed. And you can control the volume by the duration of contraction. And in this example, you can actually see how this drop is monitored and the volume of the drop is displayed here. So in this case, it's 285 picoliter drop volume. In this scheme, you see several nozzles, or actually several pictures of the same nozzle. You can see that because we use the dirty nozzle. Usually those are much cleaner. This was intended so you can actually see that it's always the same nozzle, and with just different parameters, we get a very big range of drop volumes. Now, of course, delivering a chemistry to a surface is a tough thing. So first of all, there's an interaction between the glass and your proteins. You might get a film. The whole thing might shoot a little bit sidewise. You might run into various problems. And the surface wettability of the nozzle inside and outside is a very important factor here. Also, controlling the voltage, pulse width, and the frequency will eventually lead to the stabilization of the nozzle and the drop that you get out. And eventually, you get a spot on your support, on your target, whether it's a biosensor, nitrocellulose, a usual glass slide, whatever. And in worst cases, this might look like this. And of course, if you make a microarray looking like this, I can assure you that no good results will come out. And you actually see that there's a problem in the wetting properties of the nozzle. So solution here is, for instance, to use coated nozzles. So special treatment for the outside and inside coating of the glass, which will lead to a more stable dispense and eventually to a much nicer array. And so we play around with these nozzles and surfaces, and eventually we just build them into machines which range from research and development sizes through small to medium production sizes, and at the end there's the large-scale, high-throughput production machines. The nice thing here, it's always the same technology. So if you're using the technology, or if you're using this technology in your R&D, you can just copy-paste all of your settings to a bigger machine, and you're running. So no change afterwards. It's just the same technology from start to end. And the applications are extremely various. So the traditional DNA, protein, now modern glycan arrays are actually the most used applications. Cells, living cells, so the nice thing, you can dispense living cells, which is becoming a very important thing. You can have high-throughput protein-protein interactions. You can put the whole thing into a multiplexed ELISA. So in an ELISA, you can have a 96 well plate, which eventually will give you 96 results, because each well is one result. We're not talking about taking duplicates and so on. Just take it as 96 results. With this technology, you can actually spot 96 drops into one single well, and then one patient will have one well. You're saving material, and it works much faster. So that's the intention of multiplexing. In addition, of course, to various other applications like nano-PCR and sol-gel technology, which I'll mention a little bit later. Which brings us to the delivery technology and some highlights I would like to show you. So our main aim is that we lose nothing. Well, eventually you lose nothing because the system works without any dead volume. We can work with air gap dispensing, and we can give back the rest of the probe. And so your loss, your sample loss, is zero. This is very important if you have very important antibodies that you're spotting. We don't add anything to antibodies that we spot, so no glycerol, no other additives are needed, so we just adapt our dispensing nozzle to your sample. And nothing changed, so the dispense velocity is very, well, not the velocity, the whole system is very gentle, so the dispense is very gentle, and eventually cells live after they're dispensed. So what you have been seeing now the whole time is actually a drop while it's being formed and trajected out of the nozzle and eventually forming a perfect drop. And this drop eventually goes onto your biosensor or the point of care device. By the way, the drop diameter here is around 120 micrometers. Now, these nozzles can be put in a machine, as you see here, in one dispense head, and the dispense head doesn't move, but actually the nozzles move up and down, which makes production in MTP plates much easier and much faster, because you don't need to go up with the whole head. You just shoot out your nozzles individually, and you don't run any risk that you can break one of your nozzles if you haven't correctly calculated which nozzle is where at what time. So this will make the process much faster and much flexible. One of the strengths of the technology, if you use two of these nozzles at a 90-degree angle, you can actually use two substances. If you have a polymer and a starter, you just mix them in the air, and they will be mixed by the time they hit your target, which would lie around here. And this will actually show you that the speed of the drops when they actually exit the nozzle is strong enough to cause enough mixing that you actually get a ready mixing inside your drops. So, and this dispensing speed, of course, can be adapted. Therefore, here you see a strong mixing, and here in the front, you see a much less mixing speed. Of course, the whole thing needs to be contamination-free, so each of the nozzles in a multi-nozzle setup, here we have 8 nozzles, each nozzle has a dedicated wash position, so there's no cross-contamination. And a nice fancy thing is, for instance, if you want to aspirate the residue of whatever you've been depositing, you can do that by means of the side dry. So here you see a couple of drops deposited, and the supernatant eventually sucked away. This is also important if you want to, for instance, spot on living cells. So not spot living cells, but you actually have cells in your Petri dish, and you want to spot siRNA on each cell. This is possible by the means of a head camera, so you can actually detect your cells and then spot siRNA, for instance, on individual cells. And of course, before you do that, you need to take away the cell medium, and this is what you can also do with this neat device. Spotting patterns can be extremely various. You can have one drop per spot, you can have several drops per spot, as you can see here. You can have a spot-in-spot or drop-in-drop mixing on your point-of-care device. So extremely flexible. And this goes now towards the personalized medicine. Microneedles are becoming more and more important in this field. So a microneedle, I will talk about this in more detail later on, but eventually it's a patch which has thousands of needles, usually about 100, 200 microns wide and about 400 microns high. And you see that you can actually spot on each of these needles. This is an application where you see that you see microbeads dispensed. So at a certain size of the microbeads, this is possible, which eventually also will be the same case if you dispense cells. So with a certain adaptation, this will not lead to any blocking. Now, when you have biosensors, Usually they look something like this. Very tiny, and somewhere you have a couple of golden pins where your substrate should go. So for this you need a head camera, which takes a picture of your target, and the software then analyzes where your fiducials are. So usually you have fiducials, starting points, which the camera recognizes, and then starting from these fiducials, the machine knows where to put your sample. And so this goes like we learn, or we teach the machine how it looks like. He recognizes where he should put the drops, and then deposits the drops. And the same head camera, after the machine has done the dispensing, same head camera can go back to the target and detect whether there is a drop, or there is no drop, or there is something wrong with the drop. Whether it's too big, too small, it has a satellite, whatever. So the software will detect that and give a warning. And this is very important because with this technology, you can actually make a quality control of each and every device you're producing. So you're not just picking out one of every 20 or one of 50, but you're actually checking every device that goes out to the market, which brings us to the market. So in the coming couple of slides, I will go into some applications from our customers. So the first customer to mention is the company Excella, which is Toronto-based, and they print DNA arrays and also protein arrays on diffractive optics. I had no idea what this is. I still don't. So it's a sort of chip with a nanostructure inside, and they deposit their material on top of these chips, and eventually, so these are the capture molecules, and these capture molecules capture or not capture existing or non-existing antigens from the blood, from the serum, etc. And according to whether there are molecules bound to this drop or not, the laser which goes from the side will actually be diffracted in different ways. And by the change in diffraction, they can detect or analyze whether there is a molecule bound to that antibody or antigen and how much is bound. So a very fancy test actually, and the nice thing, this is not only a closed system, this is an open system, so they will take your samples, or you can take the system, put on your samples, and then have a new detection system for whatever you're testing for. Genelite, US-based company. This is completely awkward. It's a micro-ring sensor, and light actually goes in from a small slit here and goes into that ring and emits light to the above-lying sensor. So it's like looking 90 degrees from the top. Now, if you have molecules bound here, the light will shift, so you'll get actually a shifting in your wavelength, and by that they can detect, or they can say how much molecules are bound and whether there is a molecule bound or not. And also, this is a semi-open system. They have it now as it's called Maverick, so it's for nuclear auto antibodies. And so this is already in the market, and you can see here used with several serums that the sensitivity is pretty high. Here you see how these are actually produced in the machine. So this is a spotter with this pin that you've seen inside, and they aspirate the samples from here, and here's where their targets lie. And they look like this, pretty small. They have two fiducials on each side, and this is actually the small ring, the micro ring that I've been talking about. And the software now detects the rings and spots on each of these rings a certain amount of antibody. Different sizes here are due to different concentrations, so they need to have different amounts of samples spotted on different positions. Another application is not so fancy. It's planar arrays. in microtiter plates. Genomica is a Spanish company, and they produce HPV and HPV detection systems. They have the whole hybridization and reader and so on in this small box, and produce the arrays with our technology. The nice thing here, we test the wells before and after the position, just to make sure that there is no debris inside the well which could interfere with the detection. And here you see thousands of images being analyzed and showing the accuracy of the dispensing. Diagnostics, that's the world's first cylindrical microarray. They also have a system which can be more or less adapted to your needs. They're in drug screening and sepsis, bless you. And PCL, a Korean company that makes blood testing in blood banks for HIV, HPV in a similar way to the Genomica. And this is a very fancy thing, and the last one, here you can actually configure your own test strip. So you have different small nitrocellulose pads here, which can go into a holder, different configurations possible, and then you just configure your own test. Very neat. And this is the last one, microneedles, and you see the small size of 120 microns, so it's really tiny. And with this technology, you can actually deposit exact amounts on each and every pin. Now they're just dipping it in the solution, and they're losing a lot of material, and there's no control on how much material there actually is. So I'll just skip this quickly. Hope I gave you a nice overview. If you have any questions, we can take them now, and I thank you very much for your attention.